Hi everyone, and welcome to our video on the pancreas. This is part of our Module 5 Section 4 series where we're looking at hormonal communication, and what we're going to be doing in this video is having a look at Specification Reference 5.1.4c Part 1, the histology of the pancreas, and 5.1.4c Part 2, the examination and drawing of stained sections of the pancreas to show the histology of the endocrine tissues. First thing then, let's jump into a few key facts about the pancreas that we should know. First thing, the pancreas has both endocrine and exocrine functions, and we're going to look at both of them throughout this video. What we actually find is that we have two main secretions from the pancreas. The first are hormones, and these will be secreted from the islets of Langerhans. And the second are these pancreatic juices, which contain the enzymes necessary for digestion, which are going to be secreted into the small intestine. If we start with the exocrine function first of all. Definition for our exocrine gland, remember, is a gland that secretes substances into a duct. It does not secrete them directly into the bloodstream. So what we're finding here is those digestive enzymes that we're producing are going to be passed through a duct into the digestive system. Now, the exocrine cells themselves are arranged into a little group called an axinus, and this axinus actually surrounds a small tubule in the middle. Now, if we've got many axinus, they are called the axini, and these are going to be grouped together into these small lobules which are separated by the connective tissue. So basically what we've got are a collection of these axini, and there's just connective tissue between them, but all of them are actually going to then group around to secrete their enzymes into the tubule, which then is going to end up in our digestive system. So when we actually consider what happens with all these little tubules at the centre of the axini, then they are all going to join together. And when they do so, they form an intralobular duct. So remember, intra is basically within. So this intralobular duct is then going to become the pancreatic duct. And the pancreatic duct is the one that then carries that fluid containing all those enzymes into the small intestine, or if you want to be more specific, the duodenum. That fluid that's going to be secreted into the duodenum is going to contain a selection of different enzymes. So we have lipase, which digests the lipids. We've got our pancreatic amylase, which is going to digest amylose into our maltose. We've got trypsinogen, which is an inactive protease. And then when it gets into the duodenum, it becomes the active form trypsin. We then have sodium hydrogen carbonate, which is going to make that solution alkaline, because as the stomach contents exits into that small intestine, it's clearly acidic because our stomach has a pH of around about 2.2. So what we need to do is make it alkaline, because that's then going to bring it in line with the optimum pHs for these different enzymes. The other side to the function of the pancreas is the endocrine function. And this is all to do with the islets of Langerhans. So the islets of Langerhans are actually dispersed in these small patches among those axini. Now, the islets of Langerhans themselves contain two cells that you need to know about, alpha cells and beta cells. And these are the endocrine tissues. So the alpha cells are going to be secreting glucagon, the beta cells secrete insulin. And hopefully from our GCSE knowledge, we remember that both of those hormones are involved in controlling our blood glucose levels. So what we've got in a little diagram at the bottom here is just the islets of Langerhans. So if you have a look at our little diagram, we can see that the kind of bluey coloured cells are the alpha cells, which would be secreting the glucagon, and then the greeny coloured ones are the beta cells, which would be secreting the insulin. Now, the other bits that we can see are those yellow little donut shaped things around the outside. That is actually our acini. So what we can see is you can see that the acini all surround the actual islets of Langerhans, and the islets of Langerhans made of alpha cells and beta cells. One point to note when we're writing alpha and beta on an exam paper, folks. You can write the full words, alpha and beta. You can write our little symbols, so we've got alpha and we've got beta. You cannot write just an A or a B. That is not getting you the mark. So either correct letters, so alpha, beta, or write the full words. So we need to focus on insulin and how this is actually released. We need to know the process. If we're thinking about insulin, we're talking about the beta cells in the islets of Langerhans. And the beta cells have calcium ion channels and potassium ion channels. So what we normally find is that the potassium ion channels are open, and that means that potassium ions are able to just flow out of those beta cells. In a situation where the blood glucose levels are high, what happens is glucose is just going to enter the beta cells. Now, once that glucose gets into the beta cells, it will be metabolized, and that leads to the production of ATP. When we've got ATP within those beta cells, they're going to close those potassium ion channels, and as a result of that, the potassium ions can no longer leave. Therefore, we're getting an increase in the concentration of potassium ions inside the beta cells. Because we're increasing the concentration of potassium ions, which obviously have a positive charge, then our potential difference across our cell membrane is then going to be changed, because the inside is becoming less negative, because we're obviously accumulating positive ions. So because of that potential difference across the cell membrane, the calcium ion channels open and calcium ions will enter the beta cell. When we get the increase in calcium ion concentration, that is going to cause these little vesicles inside the beta cell to undergo exocytosis, and they will then release the insulin directly into the blood. So what we do have here is a bit of a link to some of those important ions from our module 2. We're talking about exocytosis and obviously how those vesicles are going to be released. Again, dropping back to our module 2 work there. What we find then is if these beta cells are secreting insulin, they're going to continue to do that as long as blood glucose concentration remains high. Because as long as there is a high blood glucose concentration, glucose is going to be entering those beta cells, and therefore we're going to be creating the ATP, which is closing the potassium ion channels, which is then going to obviously lead to the calcium ion channels opening and the calcium ions moving in. So as long as that's happening, that is going to keep secreting the insulin. It is worth remembering that when we do have this increase in our blood glucose concentration, there is always going to be a slight delay between that increase and the secretion of insulin, because we've got to have the time for the glucose to actually get to those beta cells and pass into them. We obviously have to have that depolarization taking place, and all of that has to occur before the vesicles are able to fuse with that cell surface membrane. So because we do have a little series of steps, if you like, to work through before the insulin is released, then there's always going to be this minor delay between the increase in the blood glucose concentration and the actual release of the insulin to bring it back down into our acceptable limits. So the last thing we need to be able to do is to actually identify those key structures on a light microscope image. Now, remember, if we think back to our module 2, we need to identify how we know this is from a light microscope. And one of the easiest ways is that we can see entire cells. So that's one simple way that we can tell this is from a light microscope. Now, the bits we need to identify then, let's start off with this little region here, and that is one of the islets of Langerhans. And we've got another one down here. So both of those circled in red are the islets of Langerhans. Now, what we then find is that surrounding those, we've got these little structures called the axini, remember. So these are the axini all the way around. So all of those ones in yellow, there's obviously lots of them. I'm not going to write in yellow because you won't be able to see it off there, but those are going to be the axini. Now, what we will also find is that we do have certain other regions that we can pick out. Let me find another colour that works. So what we've got over here circled in blue, that is going to be one of the ducts that we have as well. So we can pick out those key features. The bit they're most likely to ask you to identify will of course be the islets of Langerhans, and you can generally spot them because they're going to be a little bit paler in terms of their colour, and you will be able to see them standing out quite nicely against the background of the darker purple axini there. As always, I hope you found today's video useful, and don't forget to subscribe to the channel so that you can see when the next video is uploaded, and of course head on over to the website where you can see an array of other resources to help you in your A-level biology studies.